Hieff NGS DNA selection beads operating instructions. Library purification and library sorting. Magnetic bead library purification. The required reagents are library sample, Hieff NGS DNA selection beads, absolute ethanol, sterile ultrapure water. Experiment description. This experiment takes the library sample as an example, and one sample is displayed. Library purification. Remove Hieff NGS DNA selection beads reagent from the 4 degrees Celsius freezer and equilibrate the beads for at least 30 minutes. Shake and mix the magnetic beads equilibrated at room temperature. Aspirate 50 ml of magnetic beads. The volume ratio of the sample to be purified and the volume of magnetic beads is 1:1. Blow it into a PCR tube, vortex, and mix. Incubate at room temperature for 5 minutes. The PCR tube was briefly centrifuged and placed in a magnetic stand. And the supernatant was carefully removed after the solution was clear, approximately 5 minutes. Keep the PCR tube in the magnetic stand. Add 200 ml of freshly prepared 80% ethanol to rinse the magnetic beads. Incubate at room temperature for 30 seconds. Carefully remove the supernatant. Repeat the step of adding ethanol and removing the supernatant. After that, keep the PCR tube always in the magnetic stand, and use a small pipette tip to remove the residual ethanol at the bottom of the tube. Keep the PCR tube always in the magnetic stand, and open the cap and dry the magnetic beads until cracks appear, about 3 to 5 minutes. Take the PCR tube out of the magnetic stand, add 21 ml of double distilled water. Gently mix with a pipette, and let stand at room temperature for 5 minutes. Then the PCR tubes were briefly centrifuged and placed in a magnetic stand to separate the magnetic beads and liquid. After the solution is clear, about 3 to 5 minutes, carefully transfer 20 ml of supernatant to a clean tube. Library sorting. This is the schematic diagram of library sorting operation. Take out Hieff NGS DNA selection beads Cat.NO12601 reagent from the refrigerator at 4 degrees Celsius and equilibrate for at least 30 minutes. Prepare 80% ethanol. Note, this operation takes 0.7x, 0.2x as examples, and select the appropriate ratio according to the needs in the experiment. This is recommended ratio of DNA selection beads library sorting. You can refer to this calculation. Note: when performing 2 rounds of sorting, the initial DNA sample should be supplemented to 100 ml with sterilized distilled water to reduce operating errors. Add the first round sorting magnetic beads to the PCR tube 70 ml. Vortex to mix. Incubate at room temperature for 5 minutes. Centrifuge the PCR tube briefly and place it in a magnetic stand. After the solution is clear, about 3 to 5 minutes, carefully transfer the supernatant to a clean centrifuge tube. When transferring the supernatant, generally reserve 2 ml of liquid at the bottom of the tube to avoid touching the magnetic beads. Add 20 ml of magnetic beads for the second round sorting to the supernatant. Mix by vortexing, and incubate at room temperature for 5 minutes. Centrifuge the PCR tube briefly and place it in a magnetic stand. After the solution is clear, about 5 minutes, carefully remove it. Add 200 ml of freshly prepared 80% ethanol to rinse the magnetic beads. Incubate at room temperature for 30 seconds. Remove the supernatant. Repeat the step of adding ethanol, and remove the supernatant. Keep the PCR tube in the magnetic stand, and use a small pipette tip to remove the residual ethanol at the bottom of the tube. Keep the PCR tube in the magnetic stand. Open the cap and dry the magnetic beads until cracks appear, about 3 to 5 minutes. The tube was taken out from the magnetic stand. 21 ml of double distilled water was added. Gently pipette with a pipette. And it was allowed to stand at room temperature for 5 minutes. The PCR tubes were briefly centrifuged and placed in a magnetic stand to separate the beads and liquid. After the solution is clear, carefully transfer 20 ml of supernatant to a clean tube. Note 1. When adding 80% ethanol to rinse the magnetic beads, always keep the PCR tube on the magnetic stand. 2. Add 80% ethanol and incubate at room temperature for no more than 30 seconds.
Hieff Biotechnology. To enable success of our customers, together, to make a healthier and brighter world.